So in another recording on the same Chiel Lab channel, we have gone through a visual walkthrough of how multifunctionality works in the marine mollusk Aplysia californica, because we think that that's typical of how motor systems in general are capable of being multifunctional. In this video, I'd like to briefly walk through the actual references to the technical literature in which the data for that walkthrough has been developed, so that if you yourself are interested in actually familiarizing yourself with the underlying evidence that's the basis for the statements we make there, you can do so more easily. So one of the first references, and perhaps the one that really began the field, was work done with Doug Morton, where we showed by recording in intact animals that during the ingestion phase, the grasper protracted, and it only closed during the retraction phase as it pulled food in. And then in rejection, the grasper closed as it protracted to push out inedible material. So this was the first direct evidence of the change in phase of closing relative to the protraction-retraction cycle. We then showed that retraction is induced by motor neurons, which were measured on buccal nerve 2, and that closing is induced by motor neurons measured on the radula nerves. This was all done in intact animals. And then Doug, as part of his thesis, performed intracellular recordings from closer motor neurons that were responsible for closing the grasper and from a retractor motor neuron in a semi-intact preparation. And he showed that in ingestion-like motor programs, retraction coincided with closing, whereas in rejection-like motor programs, closing preceded retraction. So this was direct measurements from motor neurons in a semi-intact preparation of the change in phase of closing and opening relative to protraction and retraction. A very important study done with Eddie Warman showed that you could extracellular record from the soma of the B4-B5 neuron in intact behaving animals. And that demonstrated that the neuron had low activity during biting, somewhat more activity in swallowing, and was intensely activated during rejection, which was our basis for trying to understand more about the role it played in rejection. We also, in collaboration with Itai Horowitz and Avi Susswein, studied the I2 muscle and showed that it actually mediates grasper protraction. Lesioning it actually disrupts protraction, and it's innervated by the B31, B32 multi-action neurons, which also have an interneuronal function, and the B61, B62 motor neurons, all of which project on a small branch of the radula nerve, which is known as the I2 nerve. We also characterized the hinged muscle, which is innervated by the B7 motor neuron, and it mediates retraction during biting, but not during swallowing. This was work that Gregory Sutton did as part of his PhD thesis. And then we also showed that during the strong protraction of biting, the grasper moves far enough forward that the jaw muscles could act to hold the grasper out and perhaps even protract it. In contrast, during swallowing, the jaw muscles act purely as retractors. The interaction between the shape of the grasper as it closes and the protractor I2 muscle was shown in this paper, because as the grasper closes, it becomes ellipsoidal in shape, lengthening the surrounding I2 protractor muscle, which increases the mechanical advantage of I2 at the peak of biting and also during the protraction phase of rejection. We also characterized and recognized that there were at least two different forms of swallowing and rejection, and during small amplitude swallows, the grasper acts purely to grasp materials as pulled into the buccal cavity. But during large amplitude swallows, the grasper can not only translate material inwards, it can also rotate it, and that means that it's playing a role in retraction as well as in purely grasping. Similarly, in rejection, we found that during small amplitude rejections, the grasper acts purely to push material out of the buccal cavity. But during large amplitude rejection, the grasper can both rotate and translate material outwards, and so it's playing a role in protraction as well as in grasping. In this paper, we show that intense B4-B5 firing delays jaw motor neuron activation, which prevents the jaw muscles from closing the grasper and pulling rejected material back in. And in this reference, we actually obtained in vivo magnetic resonance images showing that protractions are much stronger in biting and retractions are much stronger during swallowing. And this was the basis for the schematics that we show of the different behaviors. We mentioned that the anterior part of the I3 muscle has a different function than the rest of it. During protraction, when the grasper is being pushed forward during swallowing, contraction of the anterior part of the jaw muscle can hold on to seaweed as the grasper protracts through the activation of the motor neuron B38, and this directly illustrates that the I3 muscle has more than one function. It can not only be involved in retraction, but its anterior part can help during the protraction phase. This was part of Jeff McManus's PhD thesis. And then Sabrina Liu, Hui Liu, showed that firing of jaw motor neurons during biting generates almost no force, but the prior firing of these motor neurons enhances force generation when the identical motor neurons fire at higher frequencies during following. So this was part of her PhD thesis and showed the way in which neuromodulation could directly affect the motor outputs of motor neurons in the context of one behavior that must precede a second behavior. Biting, because it's a failure to grasp, must always precede a swallowing, which is successful grasping. In this reference, Catherine Chiel, as part of her PhD thesis, showed that in addition to the I7 muscle, which had been previously described by Cropper and Weiss and their associates, that there are another set of fibers right beneath the radular surface, the subradular fibers, and we were able to show by lesions in intact behaving animals that these were critical for the process of radular opening. And recently, Jeff Gill, as part of his PhD thesis, showed that in response to high mechanical loads, animals increase the firing frequency and firing duration of retractor motor neurons B6 and B9 and recruit an additional large retractor motor neuron B3. And this is then the first evidence in an intact behaving animal of the kind of size principle that's been seen in vertebrates, but at the level of individual identified motor neurons. Thus, we have been able, over the years, to accumulate a great deal of information about how a multifunctional periphery works. And again, I encourage you to look at the companion video where we provide schematic walkthroughs and show the actual recordings of an intact behaving animal. But this has given us deep insights, not just into Aplysia's multifunctional behavior, but we believe this is, in fact, how multifunctionality is likely to work in many other animals and probably in humans as well. Thank you for your attention.